Hello friends, welcome to BMH Learning. This is another tutorial in the lecture series of electrophoresis. In this video, I'm going to talk about paper electrophoresis. In the last video, we discussed about zone electrophoresis. So paper electrophoresis is a type of zone electrophoresis. If you haven't watched the zone electrophoresis video, I suggest you to go through that first. As I said, it's a type of zone electrophoresis. That means a spotting medium or stabilizing medium is used in paper electrophoresis. The role of spotting medium is played by a paper. We will talk later more about the characteristics and specificities of the paper used in paper electrophoresis. First, let's discuss about the apparatus and working of paper electrophoresis. The equipment required for electrophoresis basically consists of two items: power pack and electrophoretic cell. The power pack provides a stabilized direct current, and it has controls for both voltage and current. Now, talking about the electrophoretic cell, the cell is formed of two troughs, and this is made for a number of other purposes also. It has two electrodes, the positively charged anode and the negatively charged cathode. The two electrodes of course are connected to the power pack. Now the procedure is such that first a long strip of paper is moistened with a suitable buffer of desired pH, and then the sample is applied transversely across the central part of the strip. Then this paper strip is taken into the setup we have designed. It's placed in the electrophoretic cell in a position such that each trough gets one end of the paper strip. In order for the two ends to be perfectly dipped in buffer solution, both ends of the paper are tied with the help of certain support which keeps the ends fixed in their respective positions. Then electrophoretic buffer is poured into the troughs so that the electric current can travel through it. And then spotting transparent insulating cover is placed on this apparatus so that the experiment is not interrupted by something or is not contaminated by outside environment. After this, the power is switched on and electric field is established. Now the sample has two types of particles: the particles which are negatively charged and then other particles which possess positive charge. So the negatively charged particles would move towards positively charged electrode, which is anode. Similarly, the positively charged particles would move towards negatively charged electrode, which is cathode. Actually, it's not the individual particle which is moving. It's homogeneous group of particles which migrate as a separate band towards their respective electrode. The electrophoretic process is carried out for 16 to 18 hours. Once the time period is completed, the strip, now with the separated proteins, is taken out of the electrophoretic setup and is fixed to a solid support using a fixative such as acetone or methanol. For now, the separated proteins are not visible. So in order to make them visible, the paper strip is again placed in a container having a solution which contains a staining dye, generally bromophenol blue. After some time, you are gonna see separated proteins can be seen as distinct blue colored bands on the paper strip. Now let's talk something about paper used in paper electrophoresis. As we know, paper is the supporting material in paper electrophoresis. The paper should contain at least 95% alpha cellulose. Also, the paper should have a very slight adsorption capacity so that the proteins to be separated do not get adsorbed on the paper. The type of paper we use is a Wortmann filter paper. Now the Wortmann filter paper to be used should be of grade 3 mm. The width of the paper used should be between 2.5 to 5 cm. The paper must be having a suitable length so that both ends of the paper should touch the buffer solution. Before using the filter, it should be first washed with distilled water, which is followed by washing with 0.1 molar HCl or 0.01 molar EDTA so that the impurities can be removed. Now, on the basis of design of the instrument, paper electrophoresis is of two types: horizontal and vertical paper electrophoresis. The procedure we discussed was of horizontal setup. In case of vertical paper electrophoresis, the paper is placed vertically. Both the arrangements are equally viable, and the choice usually depends upon personal preferences. Now the technique comes with a number of applications. It is used in the separation of amino acids into acidic and basic type. The enzymes in the blood can also be separated using this technique. This is very useful technique to study sickle cell anemia in hemoglobin. The serum contains a number of proteins and they can be separated with the help of this method. This was all. Thanks for watching.